He published over 26 papers, peer review. Actually, I went and checked his publication. It's more than that, around 60. He maintained international collaboration with industry and academic centers in Middle East, Asia, Europe, and the United States. He has received several steaming awards and recognition of his work. I'm sure that you all know Dr. Hussam Zawawi. I'm sure that you remember when he actually obtained the 100 most powerful Arab under 40 Arabian business 2014, and he also achieved the 100 under 40, the world most influential young Arab in 2016. We are very honored to have Dr. Hussam Zawawi address our scientific community, and he will be talking about evidence of SARS-CoV-2 infectivity based on real-time PCR result and viral culture. Please, the mic is yours.
Thank you, Mr. Chairman, for this very humbling introduction. And I would like to express my great gratitude for the selection committee for giving me the pleasure and honor of being with you here tonight. MashaAllah, tabarakallah, Dr. Shirbini has presented eloquently and gave a beautiful overview about SARS coronavirus. So I will not repeat her work again, but I will just give you a quick background on what's happening with the current COVID-19 pandemic. Of course, as we have no vaccines so far, apparently testing is the most efficient measure to control COVID-19 and a pandemic. And hence, of course, when we are talking about outbreaks and epidemics and pandemics, test, trace, and isolate is the rule of thumb to control any kind of outbreaks, of course. And this will control the chain of transmission and will stop potentially this pandemic quite sooner. And in this regard, the General Director of the World Health Organization has said, and here I quote, "You cannot fight a fire blindfolded, and we cannot stop this pandemic if we do not know who is infected, who have a simple message for, we have a simple message for all country, test, 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 test every suspected case." And this is exactly what I would be trying to emphasize in this talk. But I think it's very important for us to know and recognize that nowadays we have potentially three tests that are available, which can help to deliver this combating strategy, which is testing. First of all, of course, which is the gold standard, as Dr. Nasreen mentioned, is the real-time reverse transcriptase polymerase chain reaction, or RT-PCR. And the second is antibody testing for COVID-19, and there's a lot of discussion nowadays with it, whether we have a machine-based testing or lateral flow-based testing as a point of care that is also currently available in the market. And there are tests that test for IgA, IgG, and the IgM, which are different immunological responses for post-infection, of course. And the last one, which is becoming quite interesting, is the antigen testing for SARS coronavirus. And I would like to leave that right to the end of the presentation. In this, throughout the talk, I will only focus about real-time PCR, because that's what's the title of this presentation. But at the end of it, I would like to touch base on the antigen testing. This figure was presented in a publication in JAMA back in April. And of course, the authors would have submitted that quite earlier than April. But that is quite debatable nowadays, of course, this figure, when it shows that RT-PCR is not effective to detect virus particles or the virus RNA 2 weeks or 1 week prior to the onset of symptoms. But it is speculated, or that's how it's demonstrated in this figure, is that PCR can potentially be effective after during the first week of onset onward, but then it drops dramatically after the third week of onset, but that's when the antibody testing becomes available and efficient to do the testing. So for us, I think it's very important for those who do not really understand how real-time PCR work. I will just give you a quick background to it. It's basically a technology that is relying on targeting the nucleic acids of any living being on planet Earth. Any cell that would have any kind of nucleic acid, whether it's DNA or RNA, would be can be amplified with the technology, which is polymerase chain reaction. With RNA-based viruses, for example, we need to have a step beforehand, which is a reverse transcriptase, in order to convert the RNA and make it a DNA. And this step is quite important here when we're talking about COVID-19, because it's an RNA virus. So reverse transcriptase is a method, or a step, I'm sorry, is utilized in order to convert the RNA to make it DNA. And then, of course, because there is a very small amount of RNA in the sample, that has to be amplified to a huge number of copies. And that is basically the job of the polymerase enzyme. And that is to try and amplify using plugs, if you would, and there are primers. Those primers are targeting certain parts of genes of interest. So, of course, we need to know the genes of interest, and of course, we need to understand and to know exactly what is the genome of the virus we're dealing with in order to design. So we personally engineer the primers in order to target the sites of interest. And then basically using other kind of fluorescent probes, they can help to visualize or quantify the amplification. And that is basically what the role of the real-time polymerase chain reaction, because that is happening in real time. Every time the virus RNA is multiplied during the PCR, it is actually quantified using the fluorescent that emerges out of the amplification. So what does that mean? I think it's very important. The take-home message of this process is that RT-PCR targets the nucleic acid, period. Regardless of the viability of the virus, whether the virus has been chopped, for example, or killed, or if it's an active virus, real-time PCR by its current status doesn't really differentiate whether this DNA has been isolated from a living virus or a virus that has died. And hence, however, there's one more thing I just want to mention before we continue, is that one of the outputs of real-time PCR is something called the CT value, or cycle threshold. Remember when I told you about the fluorescence that emerges out of the amplification? It's actually the CT value is defined as the number of cycles required for the fluorescent signal to cross the threshold. Of course, there's background noise, and the background, in order for the amount of amplified genes that to reach to a level to overtake the background noise, that is actually when the virus is quantified using the CT, and it can CT value, which can basically reflect potentially the number of cells or the number of virus particles that exist in a sample. And of course, we can semi-quantify that by developing something called standard curve. However, the CT value has actually been utilized for further utilities, because it can give quite important indications, and that is the title of this topic, of course. It can actually correlate with the virus viability. So what I would like to, throughout this presentation now, on the next few slides, I would like to take you through several studies that have assisted this hypothesis. So, but before we do so, we also need to remember one thing as well. And for those who are working with other COVID-19 patients or conducting testing for SARS coronavirus 2, I'm sure most of you do remember the prolonged positivities that you have received with some of your patients. This particular study, which was published in Clinical Infectious Disease, one of the very important journals in infectious disease domain, it actually has looked at the positivities among patients who are among 56 patients with COVID-19. And they were found to be positive for up to 5 weeks on the day of onset. And each of those patients have received 4 or 7 tests over the several weeks after the symptoms of the onset of symptoms. However, that the percentage of the overall positivities actually declines over time, where we can see that about 89% of those patients were positive in week 2, and then they continue to decline all the way to 0% in week 6. So basically, this particular study has found that 56 of those patients have got rid of the virus particles, regardless of the viability again, by testing PCR throughout the entire period. On week 6, that's when the virus was actually disappeared, and they could not detect any of the virus. But also patients have actually, the median time of symptoms onset was to negative testing was actually 24 days. So people got recovered and got rid of the symptoms quite early in comparison to, of course, other cases. But they also noticed that actually the prolonged positivity for test results can actually associate with the age and the comorbidities of patients. So, for example, patients with who are diabetic or who with older age, as well as if they have hypertension, they would have a longer prolonged for positive result. So if this is the situation now we're dealing with, when should we call that that patient is most likely has no active virus which can infect other people? And these are the studies that I'm going to present now will actually try to answer some of those questions. Although there are other exceptions, which I will cover one of them later in the talk. So, for example, this is another journal published in this. This is another article published in the same journal, which looked at 90 patients who were positive, of course, with reverse transcriptase PCR for SARS coronavirus, and every sample of those who tested positive were also cultured on virus cells, and this is the best way to test for virus viability, because if we can culture and cultivate the virus on cell lines, that is an indication. That's the only basically indication, of course, in vitro. That's the only indication that this virus is actually viable. However, if it's virus culture negative, then of course it's gonna reflect or indicate that this virus has died. So what they have found, about 29% of those cases are actually have had virus growth, but what they have found is that they never had any sample which got isolated with a virus with a CT value of more than 24. So again, for those who have conducted testing, we get to see a CT value between 10 all the way to 37 and 35 and 40 even sometimes. But what they have found in this study is that once the RCT value exceed the 24 cycles, is basically that's when anything beyond that is basically a dead virus. Another study that was published in the Journal of Clinical Microbiology, where they conducted same kind of investigation on 60 patients, however, and they could successfully isolate the virus from 12 samples that were collected from the throat, as well as 2 from sputum and 9 from nasopharyngeal swabs. And they also correlated the genome copy, which is basically through the CT value, with the virus isolation. But they found this quite different observation. If you can look at the numbers that I have circled in red, in the samples that were cultured, the CT value they had, the highest CT value they had was 35.2. However, in the other samples they could not culture, the lowest CT value was 32.9. And of course it can go up to 42, and that is quite understandable. But even to see something that crosses the 24 CT I have mentioned in the previous study, that's quite interesting, because now it did not really pass it by 2 or 3 or 5 cycles. We're talking here a difference more than 11 cycles. For those who are more visual, this is the same representation for the previous table, and you can see the difference between the cultured on the left hand side in black versus the uncultured, which is basically, or negative culture, which is basically in blue. And you can see the difference in the CT on the axis, showing the higher CT correlates actually with the non-cultured more than the samples that were cultured. Another study, I found this quite interesting because the investigators, Basil and colleagues, again published in the reputable Clinical Infectious Diseases Journal, tested quite a large number of samples or patients. They had tested 195 patients, and they collected a total of 234 samples of those patients. They found very interesting observations similar to the observation reported in the Journal of Clinical Microbiology, with a CT value of 35. They could not isolate or grow any virus out of samples that were tested with real-time PCR and gave a CT value of more than 35. And what also was beautiful here, out of all patients tested, the mean duration for the symptom onset that associated with culture positivity was 4.5 days. So patients basically ranges all the way to 18. So if we take the maximum here, patients who are 19 days since the day of symptom onset would basically test negative. So you can basically, there are now 2 different variables that we can potentially rely on in order to give us a better understanding on the virus infectivity. One is the CT value, second is the symptom of onset. And SARS-CoV-2 was significantly more likely to be isolated from samples collected from inpatients as well as ICU patients compared to outpatients respectively. I think it's very important, apart from this summary that I'm presenting here, there's another study, and it's actually a very nice systematic review that had looked at the severity and correlated that with the CT value. And what they have found is the higher the CT value, the less severe the infection is, and of course the lower the CT value, which basically reflect a higher virus load, is actually associated with more severity. And that is, of course, which means that is, and that's why patients get admitted to hospitals, to inpatients and ICU, if the virus load is quite higher, and of course that would correlate with the virus culture as well.
Dr. Hassam, can you?
A very interesting.
Can you?
I will try to do my best. I will try to do my best. Okay, this is a very interesting study. Analyzed an outbreak in Washington, And they found again a similar investigation. If you look at the figure on day 9, that was the last positive culture they had. And if you look at the lowest CT value, it was just around 35 cycles. In summary, this is a very nice systematic review. It was published only 2 days ago in the Lancet Microbe. In summary, what they have found, it's anything more than CT value of 34 is definitely will not be able to grow from what the observation they have found, as well as anything beyond 9 days of symptom onset. Again, the virus will not grow. However, just one thing, of course, we think we understand, we started to understand this virus. This is an example which shows that there's no one size fits all. So I think, of course, there are exceptions, and this is one of the exceptions. This is a brief report published in the Journal of Infectious Diseases, showed a prolonged SARS coronavirus replication. Replication means the virus viability in immunocompromised patient, and that has lasted for more than 119 days. And the patient got admitted to the hospital 3 times, got treated, and did not develop seroconversion. The last thing, this is the last slide, which is the food of thought for you. I promised you to mention the antigen testing. I think this is one of the things we will start to see more. This particular study published by colleagues at Johns Hopkins demonstrated that virus antigen can potentially be utilized to detect for active virus that are living and correlate. And this study correlated the positivity of antigen testing with virus culture, that when they did so, that has actually was comparable to the results reported by the gold standard real-time PCR. So this is potentially can be a great method when our children go back to hospital, because antigen testing is provide great results in 15 minutes. And if it's true that it can be associated with virus culture, then it's going to report the virus infectivity. And I thank you so much for giving me the opportunity to give you this quick summary. My pleasure.
Very technical, very needed, actually, presentation to our society. We have to jump right directly to the last and third presentation of this session. I'm very happy actually to present my colleague,